Hi there. In this video, we're going to focus on the second part of chapter 21, the immune system. For all living organisms, coping with pathogens is a great survival challenge. So pathogens in general are going to be environmental agents that are capable of producing disease. Now, this could include infectious organisms, but also it could include toxic chemicals, radiation, etc. So each organism is going to have three primary lines of defense that they can use to defend themselves against pathogens. The first line of defense is ultimately going to be our external barrier, which is the skin, as well as the mucous membranes, which line openings, body passages that open to the outside environment. So of course, though the skin acts as a very effective barrier to prevent pathogens from getting inside the body. However, sometimes pathogens do get past this first line of defense, and the next line of defense they're going to encounter is our second line of defense, which is going to include several non-specific defense mechanisms that will be effective against a broad range of pathogens. So I have some of these listed here or some of the cells and chemicals, etc., or physiological processes that are going to be involved in this second line of defense. But you'll have a variety of leukocytes, for example, our granulocytes, such as our neutrophils, eosinophils, and basophils. We'll also have our macrophages, and then things like inflammation and fever, which ultimately promote tissue repair and essentially promote the arrival of white blood cells to a site of infection. Finally, our third line of defense is going to be a highly specialized way of fighting against pathogens, and this is ultimately going to include some of our lymphocytes, our B cells and T cells. And these are essentially going to fight against one very, very specific pathogen, and they're also ultimately going to leave the body with a memory of that pathogen, so that in the future, if the body encounters the same pathogen, it can defeat it much, much faster. Now, collectively, our first and second lines of defense are going to be considered our innate immunity, while this third line of defense, this highly specialized way of defending ourselves, is going to be considered our adaptive immunity. Now, innate immunity is essentially going to consist of these defenses that we are born with. So it doesn't take time to develop these pathogens, as opposed to our adaptive immunity, which is going to require time to develop in response to an exposure to a specific pathogen. If we were to compare innate versus adaptive immunity, we'd see that we could kind of characterize them based on these three characteristics. So innate immunity, we can say that it's local. Now, this means that essentially the innate immune reaction, which consists of our first and second line of defense, the skin, as well as our non-specific, as well as the leukocytes that are going to fight against a broad range of pathogens, it's essentially going to occur in one particular region. So if, for example, you got a cut and bacteria was able to breach your first line of defense and get through the skin, it would encounter some of these cells locally. So you'd have local reaction, inflammation, etc., and the arrival of these non-specific white blood cells. Now, that's in contrast to our adaptive immunity, which we could say is going to be systemic. So when your adaptive immunity or your T and B cells get activated, essentially they're going to launch a system-wide attack on these very specific pathogens. Innate immunity is going to lack memory. Now, because our innate immune system is going to be effective against such a broad range of pathogens, it's not going to create a memory of any specific pathogen. So instead, every time your innate immunity or your non-specific immune system encounters this pathogen, it's going to mount the exact same response. Now, that is as opposed to your adaptive immune system, which is going to generate what are called these memory cells that are going to allow you to mount a much, much faster immune reaction to defeat a pathogen the second time it's encountered. And so I really like this little diagram over here. I included it because I think it really breaks down the two things. And we're going to go through the majority of these things that are listed over here as we move through the lecture. We'll start off by taking a look at innate or non-specific immunity. Now recall that first line of defense is essentially going to be these external barriers that are in place to help prevent pathogens from entering the body to begin with. So these, of course, are going to be the skin and the mucous membranes. Now, if you recall when we learned about the skin in 204, it consists of this stratified squamous epithelium, many, many layers of cells, and these cells are not only keratinized, but the top layers of them are actually dead. So essentially the skin creates this dead, dry, and keratinized environment that as those cells slough off, many pathogens are going to be sloughed off by that alone. But you're also going to have the secretion of sweat and sebum, both of which are essentially going to create this acid mantle. Now, this acid mantle is going to consist of lactic acid and fatty acids. And these are essentially going to create an environment where it's harder for these bacteria or pathogens to reproduce on the surface of the skin. Now, our mucous membranes, if you recall, are going to constantly secrete mucus, which is this sticky secretion, and essentially pathogens will get trapped in that mucus and get swept away by the beating cilia, for example, in the respiratory tract. There's also going to be lysozyme in the secretions on the top of the mucous membranes, and that's essentially an enzyme that can destroy bacteria by dissolving their cell wall. Now, obviously, these external barriers are not impenetrable. So if a pathogen or a microorganism gets past this external barrier, it's then going to be initially dealt with by cells of our innate immune system, or these non-specific cells. Now, in addition to our cells, we're going to see that there's also a number of antimicrobial proteins, as well as certain processes that can be triggered to help to defeat these pathogens. So in terms of cells, we'll talk about these on the coming slides, but we'll see that there's many types of cells involved in this initial innate immune response. So many of them are leukocytes, but there's also going to be a number of other types of cells that are involved in this. So a couple of them we had mentioned previously as being lymphatic cells, our dendritic, as well as our reticular cells. Many of these cells that we're going to see here that are involved in this innate immune response are going to be phagocytic. Now recall that phagocytosis is a method of engulfing and destroying a foreign cell or foreign particles, etc. So this is going to be a really important part of our innate immune reaction, and we'll talk about this a little more on the next slide. We'll also be introduced to a new type of cell, a type of lymphocyte called a natural killer cell. These are going to be an interesting cell in that they primarily destroy damaged body cells. So if, for example, a cell is infected by a virus or a cell is cancerous, we'll see that these natural killer cells can actually induce apoptosis. Now, apoptosis is essentially cell suicide. So we'll see that it's a way of inducing a particular cell to commit suicide and essentially to kill itself. Antimicrobial proteins, if you recall, when we talked about some of our leukocytes, we said that they contained these granules which had chemicals or proteins inside that when released into the environment of the tissues would actually help to defeat certain types of pathogens. So we'll talk a little more about those, as well as some other types of antimicrobial proteins that are involved in kind of coordinating the immune response and play very specific roles in destroying pathogens. Fever is going to be this general increase in body temperature. And then inflammation is essentially going to be a very localized defensive response to any sort of tissue injury. So we'll talk a little bit more about that and what causes some of the cardinal signs of inflammation, such as heat, swelling, etc. Now, before we jump into talking about the various types of cells that are involved in innate immunity, I want to take a closer look at phagocytosis because it's such an important part of the immune reaction in general. So, recall, phagocytosis is this method by which a particular cell can reach out with a cytoplasmic extension and adhere to some sort of particle or cell or antigen, etc. And then it's able to internalize whatever this debris is, this foreign particle. It's going to internalize it within the cell in this vesicle made of cell membrane. When it enters inside of the cell, it's essentially going to merge with the lysosome, and these enzymes within that lysosome are going to break down this particular substance. So let's take a closer look at what this process looks like. Hopefully that helped you to visualize a little bit how phagocytosis works. And with that, we'll take a look at some of our cells that are involved with innate immunity, starting with our monocytes, which, if you'll recall, they migrate into connective tissues throughout the body, or in some cases, they're going to migrate into lymphatic tissues or organs, and they're going to transform into macrophages. Now, macrophages, as you saw in the video, are these avidly phagocytic cells. So they are constantly phagocytizing foreign debris, foreign cells, etc. Now, throughout the body, you're going to have both wandering macrophages, and these are going to be macrophages that are mobile and are actively seeking pathogens, as well as fixed macrophages, which are going to essentially be macrophages that are stationary within a particular tissue. So for a fixed macrophage, it would not be actively phagocytic unless a pathogen actually came and moved in and came in contact with this fixed macrophage. So some examples of locations where we would find fixed macrophages are, for example, in the lungs or the liver, etc. The second type of cell that's involved in our innate immunity is going to be our neutrophils. Now, if you recall, our neutrophils are a type of granulocyte, and these are going to be specifically geared or specialized for killing bacteria. So similar to our monocytes, which can be found in high numbers in connective tissues, we'll also find neutrophils in high numbers in connective tissues. Now, neutrophils are going to be phagocytic cells, so they will be able to perform phagocytosis, engulfing and destroying foreign substances or cells. But The main way that neutrophils are going to kill bacteria is by degranulation. So recall that degranulation is going to be this process by which the neutrophils are going to release the contents of those specific granules. And this is essentially going to produce a cloud, which is often referred to as a killing zone, that's going to be very, very toxic, not only to bacteria that are in that area, but also, interestingly, to the neutrophil itself. So this killing zone of these bactericidal chemicals are actually going to kill that neutrophil as well. The next two cell types that we'll look at, which are involved with innate immunity, are again cells that we've talked about in the past. So we have our eosinophils and our basophils, both of which are leukocytes and both of which are granulocytes, meaning they have these specific granules. Now, if you recall our eosinophils, these are going to be found primarily in mucous membranes, and their function or their specific type of pathogen that they're specialized to kill are going to be allergens as well as parasites. Now, these eosinophils, which we didn't talk about before, can also perform phagocytosis. But again, we're going to have another situation where their primary means of killing, particularly because they fight against these large parasites, is going to be the release of these toxic chemicals and proteins, which can ultimately help to weaken that large parasite, which would be too large for a single cell to phagocytize. Our basophils, if you'll recall, we talked about how the basophils secrete these other chemicals that are going to help to coordinate not only to coordinate the immune response, but are going to play a role in increasing blood flow in an area, and in doing so, also increasing the mobility or the ability of these other white blood cells to enter the area and move around effectively in the area. So one new compound that we have not talked about in the past are going to be these leukotrienes. Now, these leukotrienes are going to be secreted by the basophils, and they're ultimately going to act as an attractive molecule or chemical to help to recruit other neutrophils and eosinophils to a particular area that's experiencing an active immune challenge. Now, review, we did talk about how basophils secrete histamine, which is a vasodilator, and heparin, which is going to inhibit the formation of clots. Now, again, histamine, which vasodilates, that's going to increase blood flow. And then also as a result, you're going to see this increased arrival of white blood cells coming. And heparin, which is going to inhibit the formation of clots, again, in a particular area, this is going to increase the mobility of those cells that need to get into this localized area and move around effectively to defeat that particular pathogen. The last type of cell we'll talk about that's involved with innate immunity is going to be a new cell for us, and this cell is a natural killer cell. Now, this is also a leukocyte. It's categorized under the lymphocytes, and recall when I introduced you to the lymphocytes in chapter 18, we said there was many different types and they have a wide variety of functions. And so the natural killer cell is going to be a type of lymphocyte. Now, again, this is a non-specific or a cell that is involved with non-specific immunity, and essentially what it's going to do is patrol the immune system, and it's going to be inspecting all of the cells that it comes across. So it's constantly looking out for pathogens, but specifically it's looking out for diseased host cells. So the way the natural killer cells identify a cell that needs to be killed is essentially by inspecting it and making sure that every cell it comes in contact with is displaying the normal self proteins. So each of your body cells is going to have almost like a signature tag, a number of proteins that are displayed on the cell surface that are going to be unique to you and are going to signify to your immune system that that is a self cell and a healthy cell. And so our natural killer cells are essentially going to be able to detect abnormal cells by the absence of certain proteins on the surface. When the natural killer cell detects a cell that's either abnormal or infected, such as a cell that's been infected with a virus or even a cancerous cell, it's going to release two different types of proteins that are ultimately, through their activities, going to lead to apoptosis or suicide of that infected cell. So the first type of protein that's released are called perforins. You can see in the little image here. These perforins are going to essentially poke a hole or form this little canal that is an opening or a hole in the cell. Now, that's going to inhibit that cell's ability to maintain homeostasis. And so water and ions are just going to flood through that cell membrane. And if that alone doesn't kill the cell, you'll see that the natural killer or the NK cell is also going to release these enzymes called granzymes. These granzymes are essentially going to enter the cell and they're going to break down that cell's enzymes. Now, this cumulative damage is ultimately going to induce apoptosis in this either this pathogenic cell or the body cell that's been damaged. And then ultimately, as that cell dies, you'll see that it's going to be degraded and phagocytosed by a macrophage. Okay, so to recap, we're talking about innate immunity, and we just finished talking about the cells that are involved in our innate or nonspecific immunity. The next thing we're going to focus on is the antimicrobial proteins that are involved with innate immunity. Now, these are proteins in addition to those that we saw can be released by our various types of leukocytes. Essentially, these antimicrobial proteins are going to function by either attacking microorganisms directly or hindering their ability to reproduce. And of course, depending on if the pathogen is a virus or a bacteria, this could look different in both of those instances. Essentially, however, there's going to be two families of blood-borne antimicrobial proteins. These are interferons and the complement system. So interferons are essentially going to interfere with viral replication. You can think of these almost as kind of the dying words of an infected cell. So the interferon that's released by an infected cell is not going to do anything to help the cell that released it, but it can help the neighbor cells and prevent those from either being infected or synthesizing new viral proteins, even if they have already been infected by the virus. So essentially what these are going to do is activate second messenger systems in neighboring cells that will lead to the synthesis of antiviral proteins. So again, these are not going to help the cell that's already infected, but they're going to kind of help the neighbor cells that perhaps have not yet been infected. The other family of antimicrobial proteins are going to be this complement system. Now, this is a group of over 30 proteins that are going to have a wide variety of functions and are going to contribute to both innate and adaptive immunity. So let's take a closer look at complement on the next slide. You can think of the complement system or compare the complement system to the coagulation cascade in that there's going to be all of these proteins that are circulating in an inactive form. The majority of these are going to be synthesized by the liver, and essentially what's going to happen is that they are going to become activated in the presence of a pathogen. And they're ultimately then going to activate this cascade where one enzyme activates another, et cetera, et cetera. So these complement proteins are usually named C, and then they're going to have a number after them. So you're going to see here that our main one is going to be C3. Essentially, when complement gets activated by any of these three routes of activation, which we'll talk about in just a moment, you're going to see that complement is essentially going to amplify virtually every aspect of the inflammatory process. So let's take a closer look at how complement gets activated. And then on the next slide, we'll talk about some of the effects that complement will have once it is activated. So there's essentially 3 routes of activation: the classical pathway, which is shown here, the alternative pathway, and then the lectin pathway, which are shown here. Now, the classical pathway is ultimately going to require an antibody to get started. And we'll talk a little bit more about antibodies and antigens later in the lecture, but we did touch on them in chapter 18 when we were learning about blood types. Anyhow, an antibody is specific to one type of antigen. And thus, our classical pathway is ultimately going to be part of the adaptive rather than the innate immune system. The other two, however, the alternative and the lectin pathways, are essentially part of the innate immune system because they are nonspecific and they do not require an antibody to get started. So, at this time, our focus is going to be on this alternative and our lectin pathway. And you don't need to know all of the steps of the cascade, just kind of understand how this pathway gets triggered. So the alternative pathway is going to get triggered spontaneously. And so what you'll see is that this C3 protein is going to dissociate into 3A and 3B, C3A and C3B, and this happens spontaneously. Now, C3b can bind to a pathogen, and it's ultimately going to then trigger this cascade down here. Now, note that even though this happens spontaneously, C3b is not going to bind to our own cells because our cells are going to have inhibitors for this complement process. So C3b, when it is liberated from C3, it can initiate this cascade if and when it binds to the surface of a pathogen. The lectin pathway, on the other hand, is essentially going to occur when a lectin, which is a water-soluble protein that binds sugars, when a lectin binds to a sugar or a carbohydrate that's present on the surface of a microbial cell. You can see here that all of these routes of activation are ultimately going to converge on this step of C3 dissociating into C3A and C3B. So it doesn't really matter how the pathway gets activated. Ultimately, you're going to set off this cascade that is going to amplify all of our immune responses, both in innate and adaptive immunity, which we'll focus on during the second half of this lecture. Once the complement system is activated, you're going to see that it has 4 major effects, all of which are going to contribute to the immune response. So the first of these is inflammation. Now, we're going to come back to this and talk about it in detail, but ultimately inflammation is going to be this local defensive response to any sort of tissue damage. Now, that could just be a physical injury or a breach by microorganisms. But ultimately, we'll see that complement is going to stimulate inflammation by triggering the release of inflammatory chemicals such as histamine, and also stimulating the activity of neutrophils and macrophages. We'll also see that it aids in immune clearance. Now, this is going to be the removal of foreign antigens from the blood. Ultimately, it's because it's going to bind these antigen-antibody complexes to red blood cells. As red blood cells are transported through the spleen, these phagocytes are going to remove these foreign antigens, and they're interestingly going to do so without actually damaging the red blood cells. So here we're going to see that the spleen is playing a role in this, but that ultimately this activation of C3b is going to is going to aid in the binding of those antigen-antibody complexes to red blood cells, allowing them to be transported to the spleen where they can be removed effectively. We'll also see that it increases phagocytic activity. Now, part of the way it does this is by doing something that's called opsonization. If you recall, phagocytosis is ultimately going to require these phagocytic cells to form these cytoplasmic extensions that can reach out and attach to this foreign debris, foreign cell, etc. However, depending on what that cell is trying to attach to, it can sometimes be difficult, and the microbes or pathogens can kind of evade the macrophage or the phagocytic cell attaching to them. So when a pathogen is opsonized, it's essentially going to be coated by this C3b, which is going to allow that phagocytic cell to attach to that pathogen more easily. So in that way, we're going to see that phagocytosis is enhanced. Finally, we're going to see that the complement system can actually lead to the direct lysis of pathogenic or damaged cells. So in cytolysis, you can think cyto is referring to cell and lysis means to lyse. So the way this is going to happen is we're going to see many of these complement proteins get together to form what is known as the membrane attack complex, which we'll take a closer look at on the next slide. This membrane attack complex is essentially going to be this group of activated complement proteins that are going to form a ring and ultimately a hole in the target cell membrane. Now, as we saw with the perforins earlier, which were released by our NK cells, our natural killer cells, once these membranes poke a hole in the target cell, this cell is ultimately not going to be able to maintain homeostasis anymore. So you'll see this massive influx of water into the cell, and then you'll also see that solutes now can move freely in and out through that hole in the cell membrane. So ultimately, it's this massive influx of water that usually ends up killing the cell in response to this membrane attack complex. Okay, so to recap what we've talked about so far, we've learned about the cells that are involved in innate immunity, and then we also talked about the antimicrobial proteins that are very important for innate immunity, including the interferons, and then all of the proteins that are involved in our complement system. The last part of innate immunity that we'll focus on in detail is inflammation. So recall that inflammation is going to be a local defensive response to any type of tissue injury. So this could be trauma or infection, or in some cases it could be that an infection occurs as a result of trauma. Now, inflammation, even though it kind of has a bad rap, is really going to perform some pretty important functions. So in general, it's going to limit the spread of pathogens and promote their destruction. It's going to promote the removal of debris from damaged tissue, and it's ultimately going to initiate tissue repair. Now, if you've ever experienced inflammation, which I'm sure all of us have, you know that an inflamed tissue is going to present with some cardinal signs, and these are ultimately going to include redness, swelling, heat, and pain. So on the next slide, we'll look a little bit closer at some of these specific processes or events that are triggered by the onset of inflammation, as well as what causes these cardinal signs that are associated with inflammation. I've included this flow chart here, which is kind of a diagrammatic representation of all of the events that occur as a result of tissue injury and lead to our cardinal signs of inflammation and its kind of healing effects. So what I first want you to notice is that ultimately inflammation is really going to be mediated or promoted by the release of many of these small inflammation promoting chemicals, and these collectively as a group are referred to as cytokines. Now, again, these are going to be this class of chemicals that are going to regulate inflammation and immunity, and in general, when they're released, they're going to act as paracrines or even have an autocrine effect. Now recall that paracrines and autocrines are going to be released from a tissue, and then they're going to go on locally to act on cells of that tissue, or in the case of an autocrine effector, it will act on the same cell that released it. And so as you can see, the release of these molecules, and there are many of them, are going to ultimately promote inflammation. So some of these cytokines are going to promote vasodilation, others are going to increase capillary permeability, and then even others are going to attract white blood cells to that site of damage. Ultimately, when the arterioles dilate, you're going to get an increased blood flow to that specific area. Now, this is referred to as hyperemia. This hyperemia is ultimately going to lead to the heat and redness that are associated with inflammation. So these are resulting from an increased blood flow to that area. Now, the pain that we feel that's associated with inflammation is generally going to come from either direct stimulation of those nociceptors from some of these chemicals that have been released, these cytokines, or in some cases a nerve may have been damaged directly when the tissue was damaged, or finally, in some cases, you're going to have this swelling that occurs, and this can actually put pressure on the nerves and can cause pain as well. Now, the swelling that's associated with inflammation is ultimately going to be from this increased capillary permeability that occurs. So as those capillaries become increasingly permeable, we're going to see that more fluid leaves the tissues by filtration. And ultimately, this is going to be a good thing because it'll allow for the delivery of additional white blood cells, etc. But it can also lead to swelling. And then again, this swelling can potentially put pressure on nerves and cause additional pain. The increased number of white blood cells in this localized area, again, is going to help to defeat this particular pathogen that has breached the first line of defense. And additionally, and interestingly, what you'll see is that as these capillaries become more permeable, you're going to have the leakage of clotting proteins such as fibrinogen. And recall that this fibrinogen can be converted to fibrin. And so what actually happens is you're going to get the formation of some kind of extracellular clotting around this damaged area. And that's going to help to wall the bacteria or the pathogen, whatever that pathogenic organism is. It's going to help to wall them inside of there where they're almost trapped with these white blood cells as the white blood cells are doing their work of defeating these pathogens. So ultimately, all of these effects of inflammation are going to promote healing of that tissue and clearance of that particular pathogen. Okay, so that completes our discussion of innate immunity, and we're now going to turn our attention to adaptive or specific immunity, which is ultimately going to be our third line of defense. These populations of cells that can recognize specific antigens and fight against them. Now, if you recall, we said that there was kind of three major differences between innate immunity and adaptive immunity. And those had to do with the specificity, the location, as well as the ability to generate memory. So with our adaptive immunity, we're going to see that it's systemic, meaning it's not going to be localized to one specific area. Instead, these cells that are involved in adaptive immunity are going to be circulating, and they're going to be able to fight pathogens throughout the entire system. We'll also see that it's very specific. So it's going to be directed against a very particular pathogen. Now, because of that, it not only requires an initial exposure, but it's going to take time to develop this specific immunity. So if the first line of defense is breached and a microorganism is able to enter into the body, that innate immunity, the cells that are involved, the complement system, they are always ready to fight against that pathogen. The adaptive immunity, however, is going to take time to develop. So it's going to be a slower way of defending the body than the innate immune system. However, we'll see what characteristics make it so effective as well, even though there is this little kind of downside that it takes a longer time to develop. It's also going to generate a memory of the pathogen. Now, this is going to be very important. If once you've been exposed to that pathogen once and these memory cells are created, you essentially are going to be immune to that particular pathogen in the future. So if you were to be exposed to it a second time, in general, your body is able to react so quickly that there's really no noticeable illness. And so this is ultimately how vaccines protect you from diseases. There's two major forms of adaptive immunity: cellular or cell-mediated immunity, which is going to involve our T cells, and then humoral or antibody-mediated immunity, which is going to involve our B cells. Now recall, both the T and the B cells are lymphocytes. Now, with cell-mediated immunity, you're going to have these T cells that are able to directly attack and destroy foreign cells or diseased host cells. This is in contrast to our humoral immunity, which is going to be using antibodies that are secreted by or attached to B cells that are not going to directly destroy a pathogen, but rather are going to tag this pathogen for destruction by other cells of the immune system. Now, before we jump into the specifics of both cellular and humoral immunity, we need to cover some of our basics and talk a little bit about some background information that we need to really understand both of these processes. So the first thing we're going to talk about are antigens. Now, again, we briefly talked about antigens and antibodies in chapter 18 when we went over blood types. So hopefully this feels a little bit familiar. But today we're going to take a little bit of a closer look at these antigens and antibodies and how they work specifically. So recall, an antigen is really any molecule that triggers an immune response. Not all molecules are going to trigger an immune response, right? So generally, your own cells are going to have proteins on the surface of those cells or other molecules that your body is not going to react to. And there's also a number of other very small molecules that your body comes in contact with all the time that, again, are not going to elicit an immune response. So typically, in order to elicit an immune response, a molecule is going to be large, unique, and structurally complex, right? Now, these can be proteins, polysaccharides. Now, remember, a polysaccharide is a long chain of sugars. You could have glycoproteins, which are going to be proteins that have different types of carbohydrates attached, or glycolipids, which are lipids that have carbohydrates attached. Now, the thing to think about here is that generally these molecules that elicit an immune response are going to be larger, unique, and structurally complex. So unique to that particular organism or pathogen that has that antigen. If you were to look at a particular antigen, a molecule that is capable of eliciting an immune response, you generally see that it's going to have a number of what are called epitopes present on its surface. These epitopes are also known as antigenic determinants, and essentially they're just these regions of that particular molecule that stimulates an immune response. So if you take a look at our antigen in the picture here, you can see it's this round red thing, and it has 3 different epitopes, each with a unique molecular shape. So 3 different antigenic determinants on one antigen. Now, oftentimes, or there can be times when a small molecule, right? Remember, normally to elicit an immune response, a molecule has to be large. But in some cases, you can have a small molecule that on its own would not be antigenic, meaning on its own it wouldn't stimulate an immune response. But when an individual comes in contact with it, that small molecule can bind to other molecules, and they can form a compound that becomes antigenic. Now, these molecules that are generally too small on their own but can combine with other molecules are referred to as haptens. Now, when your body is exposed to a particular antigen with each of its epitopes or antigenic determinants, this can trigger the production of antibodies that are very specific for binding to each of those epitopes. So let's take a closer look at how these antibodies work. Antibodies are AKA immunoglobulins, or you can sometimes see them abbreviated as IG. And these are essentially going to be these defensive proteins that are either secreted or can be displayed on the cell surface of certain leukocytes, and they can respond or bind to one very specific antigen. So each antibody is only going to be able to bind one antigen. So you can think that they are very, very specific. Now, the basic structural unit of an antibody is going to be an antibody monomer. So in this picture here, I'm showing an antibody monomer, and you can see that it's going to have this sort of Y or T-shaped structure. It's ultimately, each antibody monomer is ultimately going to have kind of a similar structure to antibodies, but they're each going to have a unique region called the antigen binding site that's going to give them their specificity for a particular antigen. So on each antibody monomer, you're going to have a constant region and a variable region. Now, the constant region is ultimately going to be the same for all antibodies in a specific class. Now, there are 5 different classes of antibodies, and we don't need to know the specifics of the individual classes, but you should understand that, for example, if 2 antibodies belong to the IgM family, they're going to have the same constant region. So in our image here, the purple chains and the green chains are our constant regions. So those are going to stay the same on all antibodies of that class. The variable region, on the other hand, is going to form this antigen binding site, and it's ultimately going to be unique to each antibody. And so this uniqueness is going to come from shuffling of the DNA during B cell maturation and development. So very interesting kind of side note, but ultimately it's going to come from the genetics of that particular cell that's producing the antibodies, and it's going to bind to only one specific pathogen, or one specific epitope, I should say. Now, the major type of cell that's going to be involved with adaptive immunity is going to be our lymphocytes. However, we'll also see that macrophages and our dendritic cells will also be important for adaptive immunity. Now, big picture here, the role of a lymphocyte is ultimately going to be to bind to and recognize the antigen for which it has the specific receptor or antibody for. Now, the specific receptor it expresses, or the antibody, again, is ultimately going to be determined by the DNA sequence that it's expressing to make these particular proteins. What we'll see with the T cells, though, is that even though it's their job to recognize these antigens, they oftentimes cannot do it without the help of these macrophages and dendritic cells. So we'll see that these play a very important role in activating our T cells. So the 2 types of lymphocytes that are involved with adaptive immunity are going to be our T cells, or our T lymphocytes, and our B lymphocytes, which are AKA our B cells. Now, if you recall, all of the formed elements are initially produced in the bone marrow. However, what we'll see is that our T cells are going to mature in the thymus. So you can think that T goes with thymus. And our B cells are going to stay in the bone marrow to mature. So you can think B cell for bone marrow. Now, ultimately, this lymphocyte life cycle, these lymphocytes are going to have to go through this fairly arduous training process to prepare them to function effectively in adaptive immunity. So we could think of their life cycle as roughly being this cycle of birth, training, or development, or maturation, and then deployment. So after being born, our B cells will remain in the bone marrow to mature. And our T cells, however, are going to migrate away from the bone marrow where they were born to the thymus where they will undergo their training and maturation. So let's take a closer look at this training or maturing process that the lymphocytes have to go through. So ultimately, and I really do like this picture here, so you can see that the T cells are going to migrate to the thymus, and within the thymus, they're going to undergo this very arduous training process. So ultimately, only about 2% of all the lymphocytes that are produced are going to pass what's called this positive selection and negative selection that occurs during their training process. So this testing, they're essentially going to be tested by macrophages, reticular cells, and also the epithelial cells within the thymus. Now, I'm describing this process for T cells because more is actually known about the maturation and training of the T cells than about the B cells, but authorities speculate that the process is similar for B cells, it just occurs within the bone marrow. So with our T cells, they're essentially going to go through two major stages of training, positive selection and negative selection. So during positive selection, you're essentially going to ensure that these lymphocytes, these kind of newly born lymphocytes, can recognize antigens that are presented to them. Right? So this means ultimately they have to have the proper receptors present on their surface to be able to bind to and recognize these antigens that are ultimately going to be presented to them by either macrophages or these reticular cells. If they pass that test and they can recognize antigens that are presented to them, they are referred to as immunocompetent cells. Now, after proving that they are immunocompetent, the next thing they need to do is pass a test proving that they're not going to overreact and attack the body's own cells. So they need to be able to detect foreign antigens, but they also need to be able to recognize your own body cells so they don't mount an immune response attacking your own cells. So if they've passed that negative selection test, then they are referred to as self-tolerant, and they essentially become these, what are known as these naive lymphocytes. Now, a naive lymphocyte is going to be this lymphocyte that has passed positive selection and negative selection, but it has never yet encountered a foreign antigen. It has never yet encountered the foreign antigen that it has a matching receptor for. So these naive lymphocytes then, after passing the training and maturation, are going to be deployed. So what this means is that they're going to enter out into the circulation, and they're either going to colonize lymphatic tissues and organs, or in some cases they'll stay in circulation. Okay, we have one last little topic to cover before we get to the specifics of cell-mediated and humoral immunity, and that is the function of antigen-presenting cells. So recall that our T cells cannot actually recognize, or usually cannot recognize, antigens on their own. Instead, they're going to need the help, oftentimes, of either macrophages, dendritic, or reticular cells. Now, these cells are going to be known as antigen-presenting cells, or APCs. And essentially, they're going to play the very important role of presenting this antigen or epitope to the T cell, and in doing so, they will activate that particular T cell. So ultimately, the way this works is that our antigen presenting cell, or our APC, is going to engulf an antigen either by phagocytosis or endocytosis. And similar to the normal process of phagocytosis, that antigen or that foreign debris or whatever it is going to be broken down enzymatically. And then what's going to happen is that the epitope, or the antigenic portion of that microorganism that was phagocytosed or taken into the cell, is going to be transferred out, and it's going to be displayed on the cell surface of this antigen presenting cell. And ultimately, it's going to be displayed using a specialized type of protein known as an MHC, or a major histocompatibility complex. Now, as you can see in the image, this is really just a cell surface protein that's going to hold on to that epitope, or that antigenic portion of that microorganism, and display it on its cell surface. And in doing so, this is putting it in a format that the T cell is going to be able to recognize. So let's take a look at cellular or cell-mediated immunity. Now, again, this is going to be a type of immunity in which we have these specific lymphocytes, our T cells, that are going to be able to directly attack and destroy foreign cells or diseased host cells. Now, we're going to talk about briefly the types of cells that are involved in cellular immunity, and then also the stages of cellular immunity. Note, however, that both cellular and humoral immunity occur in 3 stages. These are recognize, attack, and then finally memory. You could also perhaps help yourself to remember this by thinking of your 3 R's. So recognize, react, and remember. So there are 4 different types of T cells that are going to be involved in cellular immunity. 3 out of those 4 are only involved in adaptive or specific immunity, and then 1 type are helper T cells. These are going to be involved not only in adaptive immunity, but they're also going to play a role in non-specific immunity or innate immunity as well, due to their ability to release these cytokines that are ultimately going to kind of promote inflammation and promote the activity of our other types of white blood cells that are involved in innate immunity. So those are our helper T cells, and the other 3 types of cells that are involved with cellular immunity are going to be our cytotoxic T cells, our regulatory T cells, which are sometimes referred to as Tregs, and then our memory T cells. So our cytotoxic T cells, these are going to be our cells that actually do the direct attacking, and we'll see how it is that they attack these other cells. While our helper T cells, these are sometimes considered the most important T cells, but they're essentially going to mediate and coordinate the responses of many other types of immune cells. Our regulatory T cells, these are going to have a role in essentially helping to turn off the immune response once that pathogen has been defeated. And this is ultimately going to be very important because you don't want the immune response to carry on after the pathogen has been defeated, because it could damage your own body tissues and cells. And then finally, we have our memory cells, which are going to be responsible for maintaining this memory of this specific pathogen and allowing us to defeat that pathogen more quickly if we encounter it again in the future. Let's take a look at the first stage of cell-mediated immunity, or cellular immunity, which is recognition. Now, during recognition, you're ultimately going to have 2 things occurring. First, you're going to have antigen presentation to this particular T cell by an antigen presenting cell, and then you're also going to have T cell activation, which will follow this antigen presentation. Now, it's important to note that there are many, many, many naive T cells, and each individual T cell is only programmed to respond to a single antigen. So again, this is going to depend on the sequence of DNA that it's making these proteins from. So each individual T cell can only respond to one single antigen. So we'll see that when an antigen presenting cell comes in contact with a particular antigen, it is ultimately going to process that antigen and display it on its MHC protein. This APC will then migrate to the nearest lymph node, and while it's there, it can present this particular antigen to the naive T cells that are present in that lymphatic organ, or in the lymph node in this case. Now, only the T cell that's programmed or that is appropriate for responding to that particular antigen is going to be able to respond to that antigen presenting cell. So that is going to be called antigen recognition, the point at which the matching T cell binds to or recognizes this antigen presenting cell that contains this antigen bound to the major histocompatibility complex on its cell surface. Now, the next thing that's going to occur is an event called co-stimulation. So after the T cell recognizes that antigen, before its activation can go any further, it essentially needs to confirm that this is in fact a foreign antigen and that there is in fact a problem. And so this co-stimulation is essentially going to consist of a second protein that's going to be recognized by that T cell. And in doing so, in recognizing this second protein, it's basically a way of it double checking that this is in fact an appropriate response. So if the cell undergoes this co-stimulation, this T cell successfully completes this co-stimulation, it's then going to enter a phase called clonal selection. Now, clonal selection is going to be a process in which this particular T cell is stimulated to undergo mitosis many, many, many times, essentially producing clones or copies of itself. Now, the majority of the copies that it makes are going to go on to be effector cells, or essentially these cells that are going to go off and perform their specialized function to fight against this particular pathogen. Some of those cells will become memory cells, which we'll talk about in a couple slides. The second stage of cellular immunity or cell-mediated immunity is going to be attack. Now, this stage is going to look different depending on whether we're looking at an activated helper T cell or an activated cytotoxic T cell. Recall that our cytotoxic T cells are going to be able to fight pathogens by directly killing other cells, while our helper T cells are more going to be involved in coordinating the response of other cells and other processes that mediate, or other processes in this immune response. Now, activated cytotoxic T cells, what we're going to see is once activated, they're going to go off and essentially bind to the MHC antigen complex on any other cells that are displaying this foreign antigen. When they do, they're going to kill or cause apoptosis in that cell, essentially very similarly to what we saw happen with our natural killer cells. So once they dock and bind, they're going to release perforin and granzymes. Recall that perforin is essentially going to poke a hole in the cell, and then those granzymes are going to enter the cell and break down that cell's enzymes. And so in this way, they're ultimately going to induce apoptosis, very similarly to our NK cells, but in this case, they're doing it very specifically. They'll only be killing cells that have been infected by that particular antigen for which they have a matching receptor. We'll also see that they can release interferons as well as tumor necrosis factor. Again, interferons we've seen previously. These are going to be involved with inhibiting viral replication. And then tumor necrosis factor is going to aid in macrophage activation, and it will also be helpful in killing cancerous cells. Now, activated helper T cells are ultimately going to perform most of their specialized functions by secreting what are called these, or what are known as these interleukins. Now, there's many different types of interleukins, and each of them is going to have some different effects. However, we don't need to know the specific types of interleukins. We just kind of need to be familiar with overall how these helper T cells are going to coordinate the immune response once they become activated. So what we'll see is that, interestingly, these are T lymphocytes or T cells, which are going to be involved in both adaptive immunity. So both adaptive immunity, which is going to include this, humoral and cellular, as well as our innate immunity. And so, again, these chemicals, or these interleukins, they can attract neutrophils and then other innate immune cells to a particular site. They can attract macrophages and stimulate their activity. So here we have kind of this way of attracting other cells to a particular site. And then we'll see that it also promotes clonal selection of both B and T cells. So it's going to help to promote this mitosis of these cells, so you can increase the number of B and T cells that are available to fight against a particular pathogen. The final stage of cell-mediated immunity, or cellular immunity, is going to be memory. Now, this immune memory is going to follow our primary response. Now, our primary response in this instance is going to be the activation of those T cells after being exposed to this antigen for the first time. So our primary response is essentially mounted after the first time we've been exposed to this pathogen. Now, after those T cells are activated, whether it's a helper T or a cytotoxic T cell, some of those cells during clonal selection are going to become memory cells. These memory cells are long-lived, and they're going to also be more numerous than our naive T cells. Many of these memory cells will be found in circulation, and ultimately what we're going to see is they are going to be able to respond very quickly upon a secondary exposure to this same pathogen. Now, authorities kind of disagree on some of the reasons why they are able to mount such a quick response, but many people believe that it's just because they require fewer steps to be activated, or maybe the steps of activation are less stringent. In either case, what we do know for sure is that these cells are able to respond much, much more quickly than if you were just being exposed to this antigen for the first time. So this memory response is often referred to as the T cell recall response. And so again, when you're exposed to this pathogen a second time, these memory cells are able to launch this response so quickly that even though you've been exposed to the same pathogen, you will not show any noticeable illness or symptoms, etc. So essentially, these memory cells are able to render you immune to this particular disease. And further, oftentimes this immunity can even last a lifetime. So this is the reason why, for example, childhood vaccines are often effective for your whole life. Now we'll take a look at humoral immunity. Now, as opposed to cell-mediated immunity, humoral immunity is going to involve B lymphocytes, or B cells, rather than T cells. And unlike T cells, which can directly attack other cells and kill them, the B lymphocytes are going to produce antibodies. Now, antibodies are not going to directly destroy a pathogen or an antigen, etc. Instead, they're ultimately going to act as a little tag, kind of tagging this antigen for destruction by other cells of the immune system. So after our B cells are born and they go through this maturation and training stage, they essentially are going to become immunocompetent, and they are going to have thousands of surface receptors for only a single antigen. Now, just like our T cells, an individual B cell is only going to be specific for a single antigen. And this, again, is going to depend on the sequence of the DNA that that particular cell is expressing. So we'll walk through the steps of humoral immunity now, and recall both humoral and cell-mediated immunity, we could break them down into these three stages: recognition, attack, and memory, or we could kind of think of our three R's: recognize, react, remember. So recognition in this case is ultimately going to begin when an antigen binds to one of these antibodies that are present on the surface of a B cell. Now, when the antigen binds to these antibodies, it's ultimately going to be internalized and then redisplayed on the surface of that B cell on an MHC protein. Now, the recognition stage will continue with activation of our B cell here. So in this stage, our B cell has now internalized a specific antigen, and it's displaying it on its major histocompatibility protein. Now, if that's all that happens, that B cell is not going to go any further and become activated. Interestingly, a helper T cell must bind to that antigen MHC complex in order for that B cell to become fully activated. So when that helper T cell binds, it's going to secrete interleukins, and those interleukins are ultimately going to act on the B cell and trigger its full activation. Once that B cell has been activated by the helper T cell, it's going to undergo clonal selection, which is, again, this series of mitotic divisions. And most of those B cells are going to become what is known as plasma, or what are known as plasma cells. Some of them will also become memory cells that can be used to mount a secondary response later if you were exposed to the same pathogen again. Now, these plasma cells are ultimately going to synthesize and secrete antibodies. Now, recall, these antibodies are not going to directly destroy pathogens. Instead, they're going to essentially promote the destruction, and we'll see that they can do that in a few different ways. There are going to be 4 different ways that these antibodies can act on the antigens, either free antigens or antigens present on the cell surface of a microbe, and render them harmless. So the first of these is neutralization. So you can see that one is pictured here. And this is essentially the process whereby an antibody, which is has a binding site for an epitope, right, or this a specific part of that antigen. Essentially, it's the antibody is going to bind to that epitope or antigen, and it can mask the binding sites that it might need to, for example, enter into one of the host cells. So it can basically block certain parts of that microorganism or that pathogen, making it less effective at attacking the host cells. The second mechanism it can use is agglutination. Now, agglutination, we saw this in chapter 18 when we looked at the ABO blood types, and we saw that agglutination was this process where the antibodies would bind to the antigens and cause the cells to clump up. Now, once those cells are clumped up and being held together by those antibodies, 1, it's harder for them to invade other tissues, and 2, it's easier for them to be phagocytized. So by clumping them together, you make it easier for the immune system to locate and then also destroy them. In some cases, there can be precipitation. Now, this would be a way of clumping together soluble antigens that are not necessarily attached to whole cells. The same principle would apply here, though, where having a clump of them would make it easier to destroy them. Finally, we can see that antibodies once bound to their particular antigen, can enhance the activation of complement. So if you recall, when we talked about the complement system, we said there were 3 different ways of activating it. And the first way we talked about, we said it would be involved with adaptive immunity because it required an antibody to trigger it. So this is going to ultimately trigger complement, and then recall all of the ways that the complement system, when activated, can enhance the immune reaction, including formation of the membrane attack complex, or the MAC, which can cause cells to lyse by poking a hole in them. So the final stage then of humoral immunity is going to be our memory stage, similar to our cell-mediated immunity. Now, recall that after clonal expansion, some of our B cells and our T cells are going to become memory cells. And so our memory B cells are going to be cells that essentially are stored, that have this memory of this particular antigen. And what we'll see is that primary immune response, remember this is the first exposure to an antigen, it's going to take up to 3 to 6 days for you to really kind of prime the immune system, activate the B cells and the T cells, and for those B cells to multiply into plasma cells and start churning out these antibodies. Now, our memory cells, however, that are stored and have this memory of this particular antigen, if you're exposed to this same antigen a second time, you'll see that antibodies are actually going to appear within hours. And not only do they appear much more quickly, you actually are going to see this rapid increase of antibodies upon your second exposure. So this secondary immune response is going to be much more effective than this primary immune response. And it's ultimately going to act so quickly again that you're not even going to see symptoms of this particular illness that would be caused by that pathogen.